Good evening to one and all. Myself, Dr. Naresh Rangda, working as Associate Professor. Welcome you all in this ISFCP sponsored dialogue series. So today's speaker is Dr. Ravi Rawat. Dr. Ravi is working as an Assistant Professor at School of Health Sciences and Technology, UPES, Dehradun. He is specialized in medicinal chemistry. Uh, he is an alumni of Naipur Mohali and Birla Institute of Technology, Mishra. He is known for his expertise in the field of medicinal chemistry, synthetic organic chemistry, computer-aided drug designing, and e-learning. He qualified GPAT with All India rank 41 and Naipur JEE with rank 61. Dr. Rawat is an author, creator, and owns copyright for the software HeroMDAnalysis, Automagical MD analysis and plotting tool by Copyright Office, New Delhi. His views were featured in a Russian documentary on global perception of Russian President Vladimir Putin after the Russian-Ukraine war, produced by Izvestia Moscow. His dedication to disseminating knowledge is evident to his prolific publication record covering diverse aspects of medicinal chemistry. With this, I welcome Dr. Ravi Rawat to deliver his talk on the topic HeroMDAnalysis, facilitating GROMACS-based molecular dynamics simulation analysis. Over to you, Dr. Ravi.
Okay, thank you so much, Dr. Rangra, for introducing me, and I am also thankful to ISF Moga Punjab for actually giving me the opportunity to present my talk here. So I will be, starting my presentation. So you are already introduced by me, so thank you so much again. So this talk is actually related to my project HeroMDAnalysis. Now the topic of presentation is titled as HeroMDAnalysis, facilitating GROMACS-based molecular dynamic simulation analysis. The title can be very hard to grab if you are not familiar with the term GROMACS and not familiar with molecular dynamic simulation. But that is not an issue because I will be giving you a brief introduction to GROMACS and molecular dynamic simulation. So in short, I have created a tool titled as HeroMDAnalysis, which is actually helping in the analysis of MD simulations. So the flow of my presentation will be as I will be giving you brief introduction about MD simulations, what actually MD simulations are, what is GROMACS, which is used for MD simulations. Then I will come to my project, HeroMDAnalysis. I will also talk about the impact of my project in helping other researchers working in the same field. Followed this, I will move on to summary and conclusions, and I will also have a takeaway for undergrad students at ISF Moga, which can be helpful for them to build their career in medicinal chemistry and pharmacoinformatics. So beginning with the first question, what are MD simulations? Actually, this whole presentation can be based on one fact that if we compare how powerful are supercomputers human brain, then if we do this analysis, we find that the brain actually can do 1 exaflop calculations per second, which is actually 1 unit of measuring the efficiency of the calculations happening per second. So the unit is exaflop, doesn't matter. It is simply just 1 raised to power 18 calculations per second. But if you compare the power of human brain versus the supercomputer, then it comes out that supercomputers nowadays are actually becoming more powerful as compared to the human brain. So if we believe in this fact and we believe that we can utilize computational calculations for actually solving a lot of modern day problems, then we find that actually computers are being used in predicting and doing a lot of scientific work. So first of all, I will take you to this fact that That by computers, I do not simply mean your mobile phones and your laptops. I mean big computers which are situated in big rooms, which can efficiently do a lot of calculations faster, those which we call supercomputers, or we also call them high performance computers, right? So I'm actually comparing the efficiency of these computers, which can actually help us in solving our research problems. So the number one area where we do computational calculations is weather forecasting. We can forecast the weather for probably, let's say, next 7 days. We can forecast the weather. So this is because of the computational power that we have, which we can use in solving a lot of equations, which can actually help us to forecast the weather. Likewise, we have other examples like video games. Video games in terms are also computational equations which are getting solved. As you can that there is a lot of things happening in the video game. There is a lot of chaos which is going on. So these are actually nothing but calculations which is being done by the computer. So video game is nothing but a simulation in a sense in itself, isn't it? So another application of solving computational calculations is video games. Another example, engineering and design. We can use computers in architecture. We can do architecture with the help of computers. We can design it. We can design it for room, for a building, for a bigger space, or probably the whole city. So this is the power of computational resources available today, that we can actually simulate the whole city actually, which can help us design better cities. So another good example of using computational power. Likewise, our field of pharmaceutical sciences is not untouched. We are using computational resources for solving drug discovery problems. Like you will see in this video, we are actually simulating a small molecule called ligand with a very big protein. So this is actually a simulation of a protein and a small molecule, which we commonly call as a drug. So such kind of simulations can actually help us analyze what interactions your ligand is making with the protein. And this in turn can help us design better drugs. So these are some of the areas where computational resources are being used. So the last video that we just saw is actually called molecular dynamic simulation.
Ravi, pardon to disturb you, but your slides are not moving. Can you please check that?
The slides are not moving. Okay, sir, I will check one more time. Sir, can you see the screen now?
Yes, it's visible.
Okay. So I just discussed the simulation that we just saw is actually MD simulation, because we had a molecule and the molecules were in motion. So that is why that was dynamics, and it was a computational experiment. So that is why it was a simulation. So this is what the term molecular dynamic simulation means. Right, so we have covered the first aspect. Moving on to the second question, what is GROMACS for MD simulation? So GROMACS is just one software which is available for carrying out molecular dynamic simulations. It is actually one out of the very big list. Like we have Desmond, we have NAMD, likewise we have GROMACS. Now GROMACS is actually preferred software for carrying out MD simulation. The reason is this, that it is free, it is open source. That means you can modify the code also if you want to, because the source code is available. Apart from that, it is very flexible, as you can simulate proteins, nucleic acids, lipids, and other kind of biomolecules inside GROMACS. Apart from that, another aspect that it is popular is that it is optimized for efficiency and high performance computing. That means you can utilize the power of parallel computing and run your jobs faster with the help of GROMACS. So on this slide, I actually introduced you with GROMACS, but a key consideration is that GROMACS do not has a graphical interface. It does not have a, you cannot open this software and use it directly because there is no graphical user interface, we also call GUI. So this software is actually based on command line interface. That means you have to use a terminal and you have to actually type commands one by one in order to do your task. So MD simulation is actually a very big series of tasks. So the first step that we are doing here is protein topology, and we have to actually type the command. This is just one step out of the list of steps that we have to do. So you can expect actually that you have to do a lot of work. You have to memorize a lot of commands. You have to actually train the researcher to learn GROMACS. And still after that, the work is very, very complex and very, very manual and very, very laborious. Likewise, you can see on the screen that we are doing a solvation step of MD simulation. So command by command, we do a series of steps. So it can actually, be very tiresome. It can make the process very complex. And for a new person, it can be very, very difficult, right? So apart from that, the commands are also prone for typographical errors. By typographical errors, I mean that if you do a spelling mistake, your command won't work. If you do any kind of symbol mistake, your command won't work. Likewise, this process is also error prone. So these are some of the drawbacks of working with the command line interface of GROMACS. But I hope with this slide, I was able to make you clear that GROMACS is one software which you can use to carry out MD simulations, and it is based on command line interface. Now moving on to the aspect of our presentation, which is nothing but my own project, HeroMDAnalysis. So I started this project with the clear intention in my mind that I should make a tool which can even help an individual with no prior experience in command line interface and can do the whole task by itself. So this was the objective of my to design this tool called HeroMDAnalysis, because I believe that the command line interface task is very, very manual, very, very error prone, very, very difficult and complex, and I should convert this process into, if not automated, but semi-automated at least, and I should make the task fast, easy and simple, so that any researcher, especially those who are chemists or biologists or pharmacologists, those who are not expert in command line interface, should be able to use my tool and do the work automatically. So this was the objective. So I was actually successful in doing this task. This slide actually shows the workflow diagram, how I was able to actually create the tool. But it would be a bit difficult for you to understand in short span of time. So I will be very brief actually. So instead of being a pharmacist or a medicinal chemist, I worked as a programmer. So I used Bash shell programming as the language in which I actually wrote this tool. I used Zenity engine as the GUI development utility. I utilized shell script compiler to actually convert my source code into executable code, and my tool actually works on Linux operating system or Windows operating system with a Linux subsystem. Okay, and I'm actually distributing this tool free of cost using a website created through WordPress and file hosting service through udrop. But now let us come to the workflow, like how actually I was able to create this tool. So this actually will gonna show you actually created graphical user interface, because that was the main USP of mine to create this tool. I was able to generate raw data of all the statistical parameters by invoking GROMACS inbuilt command. Okay, so I did not actually wrote an algorithm, but I simply invoked GROMACS command through the tool itself that was actually helping me to analyze all the statistical parameters that we frequently do for MD simulations. And followed this, I actually kept one thing called plotting of raw data into HD quality images. That means the raw data that we just now generated could be converted to high quality images, which can be directly be used in your research thesis or manuscript maybe. And apart from that, I connected all these, workflows through a lot of commands actually, which includes your if-else command, your Zenity for progress bar command. For automation, I used EOF for making and creating and removing and moving directories. I used all these commands. So this is how I was actually able to, generate this tool. So this tool was actually generated with 2800 lines of code. So a lot of coding that I actually had to do. And the code looks something like this. I know the code must be, lines of code must be moving too fast, but this is just to give you a very brief that it was actually a, very lengthy code that I had to write to actually, automate the whole task of GROMACS. But if you see the working of the tool, it looks very, very simple. Let me show you. In this video, I will show you that how I can actually launch the tool and use the tool. So what I have to do, I have to simply hit launch.sh, which actually launches my tool. Simply select on to do the type of statistical parameters that I want to analyze, which includes your root mean square deviation, root mean square fluctuation, radius of gyration, the commonly used statistical parameters of MD simulation. Solvent accessible surface area, free energy of solvation, number of H-bound contacts. So you can select as many options you want to. You can select 1, 2, or many, or all, whatever you want to. So you can see the tool is now able to do all the tasks by itself without using the input from the author. So it can do the work and create the directories in which your images would be present, so that you can directly use these images in your research thesis or your research manuscript. The images look something like this. So all the statistical parameters that you selected have got their result in terms of images, which can be directly used in your research writings. So this work was actually published in Future Medicinal Chemistry, which is published by Future Sciences. The impact factor is roughly around 5, and it was published way back in 2021. So it's been 2 years that I have created this project. And apart from that, I was also able to obtain a copyright for the source code that I written for this software, because your software work is an intellectual property and it can be protected with the help of copyright for software works, right? So I was able to obtain copyright for this software work from Copyright Office of India. I'm distributing this tool free of cost from my own web portal, which is www.HeroMDAnalysis.wordpress.com. And it's been a while that I've been, since 2 years, I'm distributing this tool free of cost to the researchers. So I hope I'm able to, introduce you to MD analysis, that I was able to create a graphical user interface tool for actually analyzing the MD simulations based on GROMACS only, not any other software, just for GROMACS. Now moving on to the other aspect, like what impact I was able to create with the help of this tool. So impact of my project. So I distributed this tool free of cost to researchers around the globe, not just one country. Because of this, a bit of marketing of mine, I was able to, reach a lot of researchers in different countries. And as it has been 2 years that this project is running, there are many research works under which this tool was actually used. So roughly around 20 research works are already published, which has utilized my tool. And you can see the different nature of research works. Like some of the research work are based on natural product origin, some of them are agricultural chemist work, some of them are molecular biology kind of work, and drug discovery kind of work. So different kind of nature of work, my tool is still applicable. I have distributed it to roughly around 250 plus researchers. It has been utilized in 50 plus projects in the short span of 2 years, and I am able to gather 20 plus citations on my publication for the utility of my tool. So this is the impact that I was able to create in the last 2 years. Moving on to the summary and conclusions, I was able to develop a user-friendly tool which can be especially be useful to the chemist and the pharmacologist who are not expert with the command line interface. I'm managing this tool and giving it free of cost through my portal www.HeroMDAnalysis.wordpress.com. And in the last 2 years, there are numerous projects which has utilized my tool, actually, and have shown increased productivity analyzing GROMACS-based simulations. I have freebies to share. I am actually the a course instructor on a platform called Udemy. So I have created some of my courses. So I have free coupons to share to 2 of my courses actually. So the first course is Chemistry Structure Drawing Masterclass. So especially the undergrad or the postgrad students who are in the need of learning how to draw chemical structures, so they can join this course. I will share the free coupon of this course through Dr. Rangra, and he will be able to pass on the free coupon to the undergrad students at ISF Moga. The other course is related to calculations and fundamentals related to laboratory work, like your normality, molarity, reaction amount calculation, your percentage yield calculation. So a course is actually built on that. So again, sharing a free coupon for this course. So this way I was able to finish the whole talk, and thank you so much for your kind listening, and feel free to collaborate on any of the aspect that can be useful through any of my resources that I shared today.
So thank you, Ravi, for this wonderful talk. You have beautifully explained the concept of what is molecular dynamics, and I appreciate that you have created your own dynamic software. So on behalf of IQAC ISFCP, sir, I extend a really heart vote of thanks to Dr. Ravi Rawat, who spared time from his busy schedule to grace this event. So today we had an opportunity to hear your thoughts on the topic, that is HeroMDAnalysis, facilitating GROMACS-based molecular dynamics simulation analysis software. And this will surely be going to encourage us in our future research events. So again, thank you for this talk.
Thank you so much, sir, for giving the opportunity to present my work here.